Inside every living thing, chemical reactions are constantly occurring, enabling life. However, these reactions do not occur efficiently by themselves. They are facilitated by enzymes. Enzymes speed up reactions by millions of times, so without them, almost every biological process would take exorbitant quantities of time to occur naturally. In fact, without enzymes, these biochemical reactions would simply take too long to sustain life. But what are enzymes? Well, enzymes are a type of protein formed by a uniquely folded sequence of amino acids. These proteins are unique per specific substrate, a substrate being the substance that enzymes act upon. For example, starches and proteins are very common substrates, each requiring a unique enzyme to react with them. Once the corresponding enzyme and substrate bind to react, this temporary structure is called an enzyme-substrate complex. Another very important property of enzymes is that they can be reused and are not consumed in the reaction with the substrate. Let's now take a quick look at a sample process involving enzymes. In this specific example, the enzyme-substrate complex will be formed by an amylase enzyme and a maltose, or starch, substrate. And since each enzyme is unique to its corresponding substrate, no other substrate will be able to bind to this amylase enzyme. Currently, the most accurate or accepted model describing this binding is the induced Wnt model. The induced Wnt model describes an interaction between the corresponding enzyme and substrate as almost perfectly matching, but slightly imperfect. As the enzyme and substrate bind, the enzyme slightly modifies its shape such that it perfectly corresponds to its substrate, and the biochemical reaction is successfully sped up. It is important to note, however, that enzymes require very specific environmental properties in order to optimally function. These requirements are also unique to the specific enzyme and vary significantly across enzyme type and origin. Specifically, enzymes require an environmental pH and temperature level within their functioning range. If an enzyme's environment reaches a temperature or pH level outside of the enzyme's functioning range, the enzyme becomes very inefficient or may even become denatured, which often, but not always, means irreversible damage to the enzyme's structure. Although in the prior examples only amylase and starch complexes were shown, enzymes exist of all varieties, are everywhere, and even exist in artificial forms. In our bodies, some of the most important enzymes other than amylase are lipase and protease, which are used to break down fats and proteins, respectively. Artificial enzymes can be found in compounds such as laundry detergents in the form of a genetically modified artificial lipase used to break down fats in clothes, or in industrial and medical applications such as where fully artificial peroxidase is found in water purification processes and biofuel production. Regardless of the enzyme or application of it, however, enzymes are essential in day-to-day life, and without them, life as we know it could not exist.